afternoon, folks. Here we are from IVCC Lab, and what we're going to talk about right now, this is on page 8, we are going to talk about mitosis. And what mitosis is this quite simply cell division and the creation of new cells. The one thing that we do need to realize is that not every cell in the human body is capable of undergoing mitosis. And we'll talk about those a little bit more in lecture. But for right now, we just want to go through the 5 stages of mitosis. The first one being interphase. Interphase now sometimes is not really considered to be a part of mitosis or a stage of mitosis, but for our purposes it is. The big thing about mitosis is what we want you to realize is the cell is not actively dividing at that time. What is actually occurring during interphase is the cell is growing because everything in there is being replicated and duplicated. Most importantly, the organelles and also all of the genetic information is being copied and duplicated during interphase, but the cell is not actively dividing. So, and then the next stage is, this is where where the cell actually does start to divide, and those are the 4 remaining stages in your checklist: prophase, metaphase, anaphase, and telophase. So what we're going to do is we're going to show you the computer screen, and we have all those stages of mitosis up for your viewing pleasure. Okay, so if you want to come right around here. So when we first start looking at the stages, we have this cell right out here, and we can see a nucleus right here, but we don't see anything else. So if we look at these cells, we can see them. We can see these black structures right here. These dark black heavy structures are the chromosomes. So now during interphase, what we're seeing is we see the nucleus, but we don't see the chromosomes, because what is happening is the DNA is still packaged as chromatin. So that it would be an interphase cell. So it's pretty boring when you look at it. So here's another interphase cell up here. Here's an interphase cell right here. So we're not always, nothing too exciting to it, folks. Now we're going to move into where the cell actually starts to divide. And the first stage and when the cell is actively starting to divide is prophase. And what we see in prophase is we see the actual appearance of the chromosomes. Now in this case, though, we have to remember the chromosomes aren't in any orderly arrangement. They are just kind of clumped into the middle of the cell. But these are these dark black structures right here at the pointer showing you the chromosomes, and this is prophase. We move on to the next stage of mitosis, and that is metaphase. And the chromosomes do something very nice and very identifiable for us. They line up smack dab right in the middle of the cell. So we can see these cells, the chromosomes right here, all right in a line. So there is a metaphase cell. And then if we come over here, here is another cell in the stage of metaphase with the chromosomes lining up straight in the middle of the cell. The next stage then would be anaphase. This is when the chromosomes break apart, because remember, what is being done in an interphase cell is all of the DNA is being replicated and copied. So under normal circumstances, we have 46 chromosomes in this cell. When we come to a prophase cell, we have a total of 92 chromosomes. We have the chromosome and an exact copy of it. So when we look right here in the anaphase cell, the big key on what you're looking in an anaphase cell is there's a break among the chromosomes. 46 are going to travel this way, 46 are going to travel that way. That ensures that the new cells have the equal and the same amount of genetic information. So that is an anaphase cell. Then the last stage of mitosis is telophase. Yes, we can see the chromosomes much further apart, but the big key for telophase is this indentation that we see on both sides of the cell, and that is called the cleavage furrow. And that is what is going to end up happening is when the cleavage furrow starts to develop, that is going to extend all the way through this cell and cut this cell into 2 new cells. So one thing that we do want to make a note, a lot of times in the book says that the cleavage furrow appears in late anaphase, and that's okay. Again, there are multiple stages. Each of the stages of mitosis have about 3 stages in there. We have condensed it down. So what helps us identify the cells, because again, we're not at the PhD in cell biology yet, folks. So all I want, Mr. Fogel and I want you to remember is when you see the cleavage furrow appears during telophase for our purposes right now. The microscope slides that we showed you of mitosis, we also have some models down in the chart that we can also use. So if we come over to this model right here, here's an interphase model. Now we can also identify these 2 little black structures right here. Those are the centrioles. We did say in the last video on the organelles that they were essential for cell division, and we will come back and explain what happens to them. The one thing you can still see right here, you can see the well-defined nucleus. I will admit though, the only thing that we don't like about this is it looks like the chromosomes are appearing here, but the chromosomes actually are not appearing during interphase. We will come to prophase now, and here is a prophase cell. If we can see the centrioles now have reached opposite ends of the cell, and also we can start seeing some of these red lines coming from those. Those are called spindle fibers. What the spindle fibers do is the spindle fibers come out and attach to the chromosomes, and so when the chromosomes are ready to be split in half, the spindle fibers start to pull the chromosomes apart. But what we're trying to show you here on this model of prophase is you can see the chromosomes, but there is no orderly arrangement to how they are positioned. The next stage right here, this will be metaphase. And we can see that the chromosomes now have lined up on the center of the cell, and we can see a large number of spindle fibers coming from both centrioles and attaching themselves to the chromosomes, which is going to, and again, very shortly, once anaphase hits, the centrioles then start to pull the spindle fibers back, and that causes the break between the chromosomes. So here we go on this picture right here, on this model I mean, we can still see spindle fibers, we can still see the centrioles right here, but now if we look real closely, we do have a split among the chromosomes. And so now what is going to end up happening, folks, are the chromosomes are going to start to move apart from one another here. We come back to telophase right now, and again, what's the big structure that you're looking for or identifier? The cleavage furrow right here, this indentation, because again, as this stage continues, that cleavage furrow is going to extend from both ends and basically cut the cell off in half. We can still see spindle fibers running between. We can still see spindle fibers attached to the chromosomes, but if we notice, look at how far the chromosomes now have moved apart. Now, if we continue on to this next stage, now what we are back into 2 cells now that are back to interphase. And what we ended up, this is the end result of mitosis, and what we are looking at right here are daughter cells. This is the end result. We got a cell with 46 chromosomes here. We have a cell with 46 chromosomes here. And again, at this point, once we move back in, that we would not see any visible chromosomes. Again, the chromosomes would become stretched out into those long, fine strands of chromatin. So again, folks, we are still on page 8 of the checklist, but what we want to do is we want to go down the structures that are listed. We have already mentioned chromosomes. Okay, now the term sister chromatids, again, it's a little bit of play on words, but at the end of the day, sister chromatids are the original chromosome along with its exact copy. Okay, those are what sister chromatids are. And again, so you can see right here, this chromosome right here, and that chromosome right there, those would be sister chromatids at that point. Okay, when you can see the actual exact copy of the chromosome. Now, the next term in your checklist is a centromere. make the note, the centromere is what holds the sister chromatids together. And what I did is we drew this up on the board for you right here so you could see it. So again, here is the original chromosome with the exact copy. Those would be sister chromatids. And what is holding them together? That is the centromere. And in fact, if we even want to be more specific with the spindle fibers, the spindles of fibers attach to the centromere. So when the spindle fibers tighten up, when the centrioles start to pull them back in, the centromere is what ruptures, and that's what causes the separation of the chromosomes from one another. Spindle fibers we have already talked about. The equator or the equatorial plane, that is the area where the chromosomes line up in the middle of the cell during mitosis. So where do the chromosomes line up in metaphase? They line up on the equator of the cell or the equatorial plane. Okay, cleavage furrow we have already discussed. It's the indentation that we see in a cell that is undergoing telophase. And what is the end result of mitosis? That would be the formation of two daughter cells, right there. And then the last two terms on that page, diploid and a haploid, that's referring to the types of cells. We have two types of cells in our body, haploid cells and diploid cells. A diploid cell, if you see in parentheses, it says 2N. The N is where we put the chromosomes. So in that case, we're going to represent N as 23. So 2 times 23 is 46. That accounts for all of the body cells. Every cell in our body, with the exception of 2, have 46 chromosomes, and we can call those diploid cells. A haploid cell is a cell that contains only 23 chromosomes, and the only 2 cells in the human body that are haploid cells are the sperm cell and the egg cell. And when the sperm cell fertilizes the egg cell, we have the creation of a cell with 46 chromosomes.